Okay, I hope every piece of the technology is connected. If you don't hear me or anything, please let me know. So the next lecture is basically with the water lecture, we pretty much finished the core of kidney physiology, which basically all 4 lectures were revolving around sodium and water and how things are filtered. We went through the nephron. All we have left is 2 additional lectures where we're going to go over things that we have not yet discussed fully. Certain things, most of what we're going to discuss to some extent, I mentioned at various points, but we're going to go over largely of individual electrolytes and nutrients and other things, how they are handled. These are very important topics still, but there are a couple of smaller topics. So both this lecture and the next one is going to be pieced together with several parts. So because it's several parts, it needs an outline. So the first part will be reabsorption of nutrients. These are things we need. This is very simple. There's no complicated regulation. Nutrients are small molecules. We need them. We don't want to lose them. So basically what a kidney does is reabsorbs every last single bit of them. The other 3 smaller parts, potassium, calcium, together with phosphate and magnesium, these are small electrolytes which don't play major roles in osmosis, at least not in the extracellular fluid, but these are all electrolytes that for one reason or another, we need to keep their concentration range in a very, very narrow range. If you have too much potassium, too little potassium, it's not good for you. So we need to very tightly regulate them. Again, all of these are connected to some extent to the sodium lecture. So we will be discussing part of the nephron, which are already to some extent familiar with. So let's start with nutrients. The most important nutrient, the center of metabolism is glucose. So you have roughly 5 millimolar glucose in the blood. We already had on the sodium lecture discussed briefly how sodium is reabsorbed. Glucose is reabsorbed. It's reabsorbed together with sodium. And there's a little more detail on this slide. Instead of just one sodium glucose co-transporter, there are 2 different versions, SGLT2 and SGLT1. They're slightly different from each other in the sense that that one of them transports 1 glucose with 1 sodium, whereas the other one 2 sodium with 1 glucose. And the one that has 2 sodium can generate larger concentration gradient. So basically what you have is in the beginning part of the nephron, SGLT2 reabsorbs the majority of glucose, but it cannot generate a very high concentration gradient. So the last bits and pieces of glucose are reabsorbed in the late proximal tubule by this SGLT1 transporter. So SGLT transporters do have inhibitors. The old inhibitor, which was found more than 100 years ago, was called fluorescein. It was not a good drug because it's toxic, it's metabolized, it just wouldn't work. It was only used in experiments, but not too long ago, pharmaceutical companies developed these better inhibitors. One example is canagliflozin or Invokana, which was approved by the FDA in 2013 to treat type 2 diabetes. The idea is that if you inhibit this sodium glucose co-transporter, you're going to lose glucose in your urine. So if you're mildly diabetic, it's one way to treat it because you can just clear some glucose through the urine. It's also beneficial to some extent. If you want to lose a little bit of weight, you take it, you lose some glucose, which with it goes energy. So it basically can serve as a weight loss medicine, but you can't lose an awful lot of weight with this. And of course, it also increases dehydration to some extent because you're going to lose sodium and water together with it. And the other side effect, very logical, it can also cause urinary tract infections because if you have glucose in your urine, then bacteria have stuff to eat, so they can infect your urine easier. So that's a little bit of pharmacological relevance. This is classical old-fashioned physiology. These are the titration curves for for glucose. Let's start on the right-hand side. Somehow my pen got blocked, so I can't draw anymore. But I'm just going to use the pointer. So there are three curves here. As a function of the plasma glucose concentration, three curves are plotted. The filtered load, this is the yellow curve. Filtered load is basically the plasma concentration times GFR. It goes linearly because the more glucose you have in your blood, the more it's going to be filtered. There's no barrier for filtration. These are just large holes. So if you have 10 times more glucose, you filter 10 times more. Then you have the red curve, which is the reabsorption. Now, reabsorption through transporters, what you need to understand is that when you move glucose from one side to the other, you need a conformational change. So there's a limit of how fast the protein can flip from one side to the other to transport glucose. So once the glucose concentration becomes high enough, at one point it reaches the maximum, and then no matter how much more glucose you filter, excretion will plateau because of the transport maximum of this transporter. This is the same for enzymes. Basically, all enzymes have some transport maximum. And as a consequence of that, the green curve, the curve for excretion, stays on zero for quite a while. So normal plasma glucose is around 100 milligrams per deciliter. In that range, reabsorption and filtered load is the same. In other words, everything is reabsorbed. You don't have glucose in your urine. When plasma glucose concentration goes up, then what happens at one point, it starts to reach the maximum transport rate in some nephrons, but not others. Then you have this play. And then at one point, when the glucose reaches a transport maximum in every nephron, then this curve becomes linear. Normally, you don't have glucose in your urine. Even if you have a mild diabetes, you don't have much glucose in your urine. But for example, an untreated type 1 diabetes patient can have enormous amounts of glucose in the urine because patients in the complete lack of insulin can have extremely high glucose concentrations. And one of the issues in an early phase untreated diabetes is the enormous fluid loss, because even though 5 millimolar doesn't sound a lot, but in the urine it gets concentrated, keeps a lot of water in the urine, and basically even the name diabetes kind of diabetes means something like flow through. So basically the glucose in the urine keeps water there, and the primary symptom is large urine volumes. So if we want to express this glucose titration not as excretion, filtered load, and reabsorption, but as the clearance of glucose, you can see here that the clearance of glucose is zero in the normal range because you don't have glucose in the urine. But then at one point, the clearance of glucose starts to increase. And just as a reference point, clearance of inulin, which is the same as the glomerular filtration rate, is up here. So you can see that at very high glucose concentrations, the glucose clearance can actually get relatively close to the GFR. So you can clear an enormous amount of glucose by this mechanism. So this slide shows the reabsorption mechanism for amino acids and carboxylates. So amino acids are also very important nutrients. This slide is a lot more complicated than the glucose mechanism, because glucose you only have one, versus amino acids you have 20 of them. So for that reason, you have many different mechanisms, many different transporters. You don't have to worry about memorizing all these transporters. The idea is that all amino acids are reabsorbed either by a sodium coupled transporter, just like glucose, and some of them are reabsorbed by a proton-driven one, because if you remember, this is the proximal tubule where you have significant proton secretion, and the proton gradient can also be used to reabsorb amino acids. And as the end result, the proximal tubule will reabsorb all amino acids normally. If somebody has mutations in some of these transporters, they can have diseases which cause amino aciduria. We're not going to discuss in details what those are, but there are some of these rare diseases where people excrete amino acids in the urine because some of these transporters don't work. So on the right-hand side, you can see these carboxylates. So you don't only have glucose in your blood, you also have lactic acid, pyruvic acid, acetoacetic acid, which are also nutrients, and these can be reabsorbed again with sodium chloride transporters. Again, the idea here is that all of it is reabsorbed normally, except if you have type 1 diabetes, when you don't only have enormous amounts of glucose in your blood, but you can also have ketoacidosis with a lot of lactate and acetoacetate. And in that case, diabetic patients can also have ketoaciduria. But normally, again, the concentration of these are relatively low, so they're fully reabsorbed. So one more slide about nutrients. So normally you have amino acids in the blood, but you also have small peptides in the blood. And some of those small peptides are also reabsorbed via these oligopeptide proton co-transporters. And even though I always emphasize at the very beginning that the primary filtrate is protein-free plasma, that being said, there's always a little bit of protein in the urine, in the filtrate, and normally that's very low concentration. And normally all of the protein from the primary filtrate is also reabsorbed by endocytosis. Proteins are too large to move through transporters, so it's easier to reabsorb them by basically just phagocytosing from the tubule. So that was nutrients. Any questions on this topic? So now let's go to potassium balance. So potassium is one of those electrolytes where you have to keep the concentration in a very narrow range. And the reason for that is explained on this slide. You had, from cellular physiology, you must have learned about the determinants of the membrane potential. Sounds like an esoteric topic, but it explains very well why too much potassium is very problematic. And basically, this equation here is the Goldman-Hodgkin-Katz equation. It shows what the determinants of the membrane potential are. And you can see that there's potassium outside and inside, sodium outside and inside, chloride inverted. And in each case, the concentration of the ion is multiplied by its permeability. And so you can imagine that something that has a very high permeability, like potassium, has an outsized influence on the membrane potential. And normally, most cells are more permeable to potassium than any other ion. And therefore, how much potassium you have in the extracellular fluid is a very important determinant of the basal membrane potential. Explaining it in somewhat simple terms, if you have too much potassium, then your resting membrane potential is going to go closer to zero, which is also called depolarization. And if you have too little potassium, then the membrane potential is going to be more negative. It's also called hyperpolarization. And so both high and low potassium is problematic because it moves your resting membrane potential to either the depolarizing or the hyperpolarizing direction. And the organ where it causes the most trouble is your heart. Your heart has to beat. It can't stop. It can't stop. And this slide on the right just shows you EKG signals at different potassium levels. You don't have to worry about the details of this in the kidney physiology lecture, but you can see that especially if you get very depolarized or very high potassium, you can get ventricular fibrillation because the increased resting membrane potential just simply messes up the electrical signal generation in the heart. So potassium is very dangerous because basically by doubling the concentration in the blood, which causes zero effect on osmolarity, by doubling the concentration of the potassium in the blood, you can have life-threatening arrhythmias. If you go to the other direction, I think it's less dangerous from the standpoint of the heart, but it causes all sorts of hypoexcitability issues like muscle weakness and constipation. So what you also need to know about the potassium balance is that much of the potassium in your body is in the intracellular fluid. If you remember from lecture 1, the sodium potassium ATPase moves all the potassium to inside of the cells. You have a ton of potassium inside, almost very little potassium outside. And this has a very high importance because we can use the high intracellular potassium concentration to essentially move potassium inside the cells, avoiding trouble in the extracellular fluid. The issue with potassium is that many of the foods that we eat has a lot of potassium, because potassium is not only inside of our cells, but inside of every cell. So if you drink an orange juice that was pushed out of oranges, and it contains a lot of potassium, same with bananas, meat, you name it. And so the issue is, if you just make a very simple calculation, the fact that orange juice has 50 millimole per liter potassium, so one glass has 10 millimole, if you play with the numbers just a little bit, you realize that 7 glasses of orange juice, if you drink it, and if it reabsorb immediately from your gastrointestinal tract, it could double your potassium, and they just told you that that can kill you. So, and of course, 7 glasses of orange juice doesn't kill a normal person. My kids would not have grown up if that was the case, and you don't see big warning signs on orange juice that don't drink more than half a glass. So the body really regulates potassium very strictly. And there are quick and slow mechanisms. The issue is if you drink 7 glasses of orange juice, there's no way the kidney can excrete it fast enough. So, so there is a rapid mechanism, which is basically increased cellular uptake of potassium that averts quick, large changes in plasma potassium. And the slow mechanism by excreting by the kidneys is the one that actually maintains the long-term potassium balance. So here is a slide essentially explaining this and giving you some details of how potassium is moved to inside the cell. This is like a graph when you drink a lot of orange juice or eat a lot of bananas. So you can see that plasma potassium jumps up a little bit, then quickly goes back to normal. And you see this very, very slow decrease here. And the bottom graph shows that much of this initial load very quickly goes to inside the cells. And once it reached maximum, so this big peak is gone, then from inside the cell, it slowly comes back outside, and the kidneys slowly excrete the whole thing. So coming back to the Goldman-Hodgkin-Cox equation and membrane potential, the reason why moving potassium inside is not an issue is because your concentration inside is very high. So if you move 5 millimolar to inside where you had 140, you move from 140 to 145, that's a negligible change in terms of percentages. So it's not going to have a major effect on membrane potential. There are 3 factors that significantly influence this uptake. The first one is passive. It just happens so that the sodium potassium ATPase, the KD or KM, or the half maximum activity, is close to the plasma potassium concentration. So if you increase plasma potassium just by passive, simple enzymatic effect, the enzymatic activity go up, whereas if the potassium is low, it's going to go down. So without any hormonal involvement or any fancy regulation, the potassium ATPase itself is sensitive to potassium concentration. That's one effect. But there are 2 additional ones. One of them is that insulin stimulates the sodium potassium ATPase. So what's the point of it? How does insulin come to this picture? Why is it insulin? Why not something else? Yes. Once again? But that's glucose. That's not potassium. So yes.
When you're eating increased blood glucose and
Exactly. So first when I saw it, it was like, what is this? But then if you think about it, what else is in orange juice? Glucose, right? Or not glucose, sugar. So the bottom line is much of the potassium load comes when you eat. And the primary hormone that clears out glucose from your blood is insulin. And the body just uses the exact same hormone to clear out potassium from what you eat also, because most of the food contains significant amount of potassium. So that's insulin. And the other one is epinephrine, which is the sympathetic neurotransmitter. And here the idea is that when you do physical activity, potassium tends to leak out of your muscle cells. And whenever you have high physical activity, you also have high sympathetic activity. So the epinephrine is another reason, another mechanism which helps keep potassium levels in normal range, mainly upon physical activity. And these two, insulin and epinephrine, can actually be used clinically. So if somebody is hyperkalemic, too much potassium, one way to treat it is infuse insulin plus glucose together. You don't want to make somebody hypoglycemic, but if you add insulin and glucose together, that can rapidly decrease plasma potassium. And the other medication that people use is beta-2 receptors are also used, beta-2 agonists for asthma. So the same medication that people use to asthma to dilate your bronchi, the same medication can actually also be used to decrease your plasma potassium concentration for these physiological reasons. So we sort of deviated from the kidney a little bit because it's also part of how we regulate potassium. But now we actually come to the kidney. So with kidney regulation, what you need to know is that that the proximal tubule reabsorbs 2/3 of potassium, just like for many other electrolytes. The problem with potassium is the thick ascending limb, this furosemide sensitive cotransporter, reabsorbs an additional 30%. So overall, these two segments reabsorb 95%. And that's an issue because that's more reabsorption than what you need. And it makes it very difficult to get rid of potassium. And the way around that is that the kidney, after reabsorb much of potassium, the kidney in later nephron segments in the principal cells will also secrete potassium. Again, we had this cell type before, the principal cell for sodium regulation. Basically what happens here is sodium is reabsorbed, potassium is secreted. So basically, because of the sodium reabsorbing mechanisms, because this guy, the sodium potassium 2-chloride cotransporter, needs potassium to reabsorb sodium, the side effect of that is very little sodium left in the lumen, but later segments actually compensate for that by secreting additional potassium. And in the case of potassium, the secretion will be the regulated step. So passive regulation of potassium secretion is that increased plasma potassium stimulates the sodium potassium ATPase, not only in somatic cells, but also in these principal cells. And if a cell takes up more potassium, it's going to also be able to secrete more potassium. So that's one simple passive mechanism. Another mechanism is it's not really regulation, but things that that increase tubular sodium concentration or generate high flow rate. Both of these will increase potassium excretion. So if you have high tubular sodium, that will increase sodium entry, which will depolarize the cell. So that will be a driving force for potassium exit. And if you have high flow rate, what happens is you wash out potassium. There's less potassium here. It's easier to secrete it. And the reason why these passive influencing factors are important to know is because two different diuretics, loop diuretics and thiazides, increase potassium loss partly by this mechanism. And in the case of loop diuretics, they also inhibit a potassium reabsorbing mechanism, but thiazides don't do anything directly to potassium, but since they both stimulate sodium concentration and stimulate flow rate, therefore thiazides also lead to potassium loss. And again, coming back a little bit to diuretics, if you use, for example, amiloride or an aldosterone antagonist, which acts directly on this principal cell, these will have an opposite effect on potassium. These will decrease the secretion of potassium. So these are the so-called potassium-sparing diuretics. So basically, these passive effects explain why certain diuretics increase and certain diuretics decrease potassium secretion. And now we get to the hormonal regulation of potassium secretion. Again, this is more of a review of what we had before already, because secretion happens by the principal cell, and the primary regulator of the principal cell is aldosterone. So aldosterone doesn't only reabsorb sodium, it also secretes potassium. And so the cell type that makes aldosterone is the glomerulosa cell in the adrenal cortex, is not only responsive to angiotensin II, but it's also a sensitive potassium sensor. So aldosterone is a key regulator of potassium secretion. That concludes potassium. If you have any questions about potassium, you can ask now or you can email me. If we are okay, then let's go to the next one. So calcium balance. So calcium is very similar in a way to potassium, because if you have, especially if you have too little of it, it can be very dangerous, because low calcium can induce hyperreflexia. You can get these strange muscle contractions. And if your hand looks like this, it's not that dangerous unless you're rock climbing or something. But what makes it really dangerous is that your laryngeal muscles can construct, and it can basically suffocate you. So hypocalcemia is very dangerous. You want to avoid it at any cost. Hypercalcemia is also problematic. You have this mix of symptoms: bones, stones, groans, and moans. It's an old way to remember it. And so when we look at calcium in the plasma, calcium is found in half free form and half bound to mainly plasma albumin. What is important in determining the excitability is the ionized calcium concentration. And once calcium is filtered, 2/3 of it is reabsorbed in the proximal tubule, and the rest of the calcium reabsorption happens in the thick ascending limb, which we already discussed the mechanism of. I'm going to review it in the next slide. And there's an additional mechanism in the distal convoluted tubule where an additional 8% is reabsorbed. And the major regulatory hormone is parathyroid hormone, which you will hear more about at the endocrine lectures. And at the end, more or less 0.5% of the filtered load is excreted. So these are the two mechanisms of calcium reabsorption. The thick ascending limb we already discussed. This is where you have the sodium potassium 2 chloride cotransporter and the counter movement of potassium, which generates the positive lumen. And the positive lumen is a key driving force for calcium, sodium, potassium, and magnesium reabsorption. Among these, really, calcium and magnesium which is quantitatively important, basically. And this is one way for reabsorbing calcium. And the other way is a classical transcellular movement or mechanism where you have an apical calcium channel. It's called TRPV5. You don't have to remember the name. It goes through this calcium channel and enters the cell, binds this calcium binding protein called calbindin, and then it will be pumped out on the basolateral side, either by the plasma membrane calcium ATPase or the sodium calcium exchanger. Again, this is a classical vectorial epithelial transport. Calcium goes in on the apical side and gets pumped out on the basolateral side. So the regulation of calcium reabsorption is mainly done by parathyroid hormone. Parathyroid hormone, or PTH, you will learn in more detail at the endocrine physiology lectures. Here I only mentioned the key parts to understand the kidney. It's secreted in the parathyroid gland in response to low plasma calcium concentration. Low plasma calcium is sensed by this G protein coupled receptor, which is called the calcium sensing receptor. And so basically, if you have low calcium, then PTH is secreted. It will act through cyclic AMP, and it will increase calcium reabsorption in the kidney, both by stimulating this epithelial calcium channel and in terms of expression, and also it has acute effects through cyclic AMP. Stimulating reabsorption in the kidney is not nearly fast enough to get you out of hypocalcemia. So even though this is a kidney lecture, this one I listed first, in terms of overall physiology, when you become hypocalcemic, the major effect of parathyroid hormone is to release calcium from, it's written up there. Where can you release a lot of calcium quickly if you want it? Your bones. Your bones are, the reason why bones are hard, it's a calcium phosphate complicated something, but you have literally kilograms of calcium in your bones. So if you release half a gram, it's not going to make a big difference from your bones, but it can save your life by moving back calcium to the normal range. And it also stimulates active vitamin D secretion, which will be important in intestinal calcium absorption. Again, you will hear about active vitamin D3 at the endocrine lectures and the last slides of the next lecture, where we discuss the role of kidney in endocrinology, because it also happens to be generated in the kidney, even though the final big effect of active vitamin D3 will be in the intestines. And as you will learn from endocrine physiology, and also from the next slide in phosphate reabsorption, parathyroid hormone has an opposite effect on phosphate reabsorption. So parathyroid hormone increases calcium reabsorption and inhibits phosphate reabsorption. The parathyroid hormone regulates calcium and phosphate in an opposite way. You will again learn the details in more details in endocrine physiology. That was calcium. Now we come to phosphate, which is also found both in bound and free form, except that it's mainly bound to other small solutes, including calcium, but it's also bound to proteins, but to a lesser extent. And the key mechanism of reabsorbing phosphate is the sodium phosphate cotransporter in the proximal tubule. And the phosphate that it's excreted that will end up in your urine is roughly 10% of what was filtered. It's part of titratable acids, which we will discuss in the next lecture. And again, as I said before, parathyroid hormone inhibits phosphate reabsorption by inhibiting this sodium phosphate cotransporter. And active vitamin D3, on the other hand, promotes phosphate reabsorption. So you will learn this in endocrine physiology, that parathyroid hormone moves calcium and phosphate to opposite directions, whereas vitamin D3 moves them in the same direction. So the combination of these two guys can actually regulate the two ions overall. I wouldn't say independently of each other, but separately from each other. But it's a little bit of a complicated crosstalk, but it's just the way it is. And finally, magnesium transport. So magnesium is a little bit similar to calcium. If you have too little of it, you get hyperexcitability. If you have too much of it, you can have cardiac effects. I think the effects are not as dramatic as with calcium, but similar in terms of direction. So magnesium reabsorption is strange because only 15% is reabsorbed in the proximal tubule. The vast majority, 70%, is reabsorbed in the thick ascending limb with the same paracellular mechanism as calcium, and 10% is reabsorbed in the distal convoluted tubule. And for the mechanisms, I actually brought in slides from pathophysiology, where you can see that that this just depicts the thick ascending limb in slightly different fashion than before. Again, it's the furosemide sensitive cotransporter, potassium out, lumen is positive, drives magnesium and calcium. And what's here, in addition to what I already showed you, is this paracellular transport mechanism for both magnesium and calcium goes through these claudin proteins, claudin 16 and 19. And when these are mutated, you get this disease called familial hypomagnesemia with hypercalciuria and nephrocalcinosis. Basically, what happens if these claudin proteins are mutated, they get less permeable to magnesium and calcium. You end up with magnesium and calcium in your urine. Calcium in the urine can cause kidney stones, and of course you get hypomagnesiemic and to some extent probably also hypocalcemic, even though the calcium is probably more tightly regulated than magnesium. So I think the primary symptom is hypomagnesemia. And on the bottom slide, you have an additional mechanism that in the distal convoluted tubules, you also have this magnesium permeable channel called TRPM6. You don't have to remember the name, but if this TRPM6 channel is mutated, again, you get hypomagnesemia, because this transcellular path goes slower. So that these two human mutations actually tell you the two different molecular mechanisms of how magnesium is reabsorbed in the kidneys. And end of this lecture. This was shorter, and the next lecture will be also shorter. Hopefully we can finish on time. If you have any questions, you can ask now. But let's take a 5-minute break to make sure we end by 5.
Professor?